I'm John Gribben, and we're here at the end of day 2 of another very successful international workshop on non-Hodgkin lymphoma 2018. Our last day in sunny Nice before returning back to think about everything we've learned here and start putting it into practice. I'm joined by my co-chair, Anas Younes from Memorial Sloan Kettering, Caron Jacobson from the Dana-Farber at Harvard, who runs the CAR T-cell programs there. So another very nice day. So you chaired, Anas, the first session on precision medicine. And of course, we've all got very good ideas of what precision medicine means, but actually it's quite hard to articulate and put into words exactly what that means nowadays. There's a kind of feeling often that the panels and the information we can have from our patients has gone ahead of what we're actually able to do about it. So you see patients all the time at Memorial who've got this whole kind of panel of things. What kind of conversations does that lead for you to have to your patients about how you talk about applying precision medicines in the way that we were talking about in the session this morning?
So it is frustrating if you're a patient to know that whatever treatment we give them, it's hit and miss. You know, we say, 60% chance you may respond.
But then it's even worse when you've got a piece of information that says it's even more miss than that.
That's
right?
correct. It could be even lower response rate than that. But what I tell my patients, this is statistics. For the individual patient, you're either 100% response rate or 0% response rate. That's what it is. There's no 60% response rate for each patient, right? So we're trying to increase the chances of you are going to be among the 100% responders than not. That's what basically is in a nutshell. But the reason why we had this discussion is mainly because the multiple failures of clinical trials that you and I and you know about, that the standard R-CHOP plus drug X has failed over and over and over again. And the question, should we come up with different strategies? Should we use more precise biomarkers? Should we use R-CHOP plus drug X in a more defined group of patients? Or should we now move to a new strategy, which is R-CHOP plus followed by drug X, based on using circulating tumor DNA?
Sure. And in terms of what that drug should be, you'd then, I liked your approach of different kind of baskets of trials, of being directed by biomarkers as to which particular direction a patient could go into. Do you think the regulators are going to buy that kind of a process, or do you think we're going to end up having to use drugs that have somehow gone through some other kind of approval process to be able to get access to them?
So there's precedent for approval, right? iSpy for breast cancer, they got approval based on this design. This, of course, is challenging. Not every bucket is expected to lead to drug approval. But yes, there is precedent for this, and we're hoping that some of these, for us, I think this master lymph protocol that we're talking about, to me, the way I see it, this is for screening, not for drug approval. If we find a doublet, let's say, that works in a unique, well-defined biomarker driven way, I think you can hand it over then to whoever, sponsor or CTEP, whoever want to confirm it to the trial to lead to drug approval. I don't think our job is to get drug approved. We're screening, basically.
Yeah. But of course, it's like everything else. Without the approval, how do we get our patients to get access to that drug? I guess also thinking about the patients that you land up taking to CARs, there is, of course, this question of bridging them safely and getting the patient through that transition, of getting them into your hands and through that process of doing it. Was there anything you heard on the next section that we talked about, either precision medicine there or in the novel drugs, that you thought, oh gosh, that's a kind of useful approach that could be beneficial?
Yeah, I mean, so I think there's clearly some patients whose disease can be stabilized or who can respond to drugs like Ibrutinib, and there's potentially even a beneficial effect in that setting of pre-treating someone with Ibrutinib and then giving them their CAR T-cells back so that the T-cells may even work better. But something that was very intriguing is the high response rate and the depth of response that we're seeing with Polatuzumab combinations, the anti-CD79 conjugated antibody. And I think that may offer something for a traditionally chemo-insensitive group of patients to debulk their tumors, to make it not only the CAR T-cells not only more effective, but potentially safer as well.
What I didn't know, and it didn't come out from Laurie's talk on the Pola, was how much CD79 depletes also normal B cells. So in other words, would a patient being bridged to a CAR T-cell approach with Pola lose some of the cells that drive the proliferation that you perhaps need of the CAR T cells once they're in the body?
I mean, these patients have also had a lot of Rituxan as well. I mean, at this point, maybe they're CD20 negative B cells that are still hanging around, but we give a lot of B cell depleting therapy in its stead. So, I'm not sure if that's going to be as important, but this is why I think that in terms of picking a good bridging therapy, it really does need to be studied in a formal manner. I think right now people are doing a lot of hodgepodge things, and we have no idea what we're doing to the chances of the T cells expanding, to ultimately toxicity or efficacy in these patients.
Yeah, but it didn't occur to me to ask the question until after Steve Schuster raised it in the CAR T cell section, which you chaired, about the whole issue of whether any of these types of therapies actually impair our ability to collect T cells for a CAR T cell patient. I didn't ask that question as we're going through the section. And of course, I guess we've got a very good sense of what happens to the therapies we use, but it's a big unknown if you're using an unknown drug, what impact it may have on that T cell population.
I mean, I think right now we don't really know anything about the T cells we're collecting from the patients or the products that we're giving back.
It's a bit of a black box.
Yeah, I think we have a little bit of insight from Juno in terms of their defined CD4 to CD8 ratio, but the other companies are giving, they're giving whatever T cell product back to the patient. I think, we all are interested in, they have some of that T cell product, and we should be able to learn more about what the composition of that product is, and whether there's a certain ratio of different immunophenotypes of T cells that are given back to patients that are associated with better efficacy and less toxicity. So I think that's going to be a big move, and something that comes out in the short term, because I think that's really definable. And then I think also, the numbers looking at prior therapy, what the prior therapies are very small, but people are looking now at, how did patients do following Ibrutinib prior therapy? How did they do following lenalidomide prior therapy? Different drugs that we know have immunomodulatory properties, to see if there's any improvement in T cell health or detriment of T cell health in terms of the product that's collected.
And of course, now George Deng stood up and asked a question which I hadn't thought of before either, is as CAR T cells move earlier forwards, as we're clearly seeing that they're doing, and everyone's excited that that's the way forward and has curative potential, that it actually decreases the pool that he can have to have patients enrolled in other studies. Now, I think Steve Schuster said, so what? But because we've got a better treatment, that's the new issue. But is that your experience? You are seeing patients earlier and fitter now than you did in the past, or is it still pretty much the train wrecks that are arriving, referred in from elsewhere, coming to your institutions for CAR T cells?
I think as a rule, people are identifying patients earlier. So especially patients who either are refractory to upfront therapy or who relapse early, people, referring physicians know at that point that that's likely going to be someone who may end up getting CAR T cells. And as a result, they're sending them in ahead of second-line therapy, both because there are now second-line trials for those patients, and they're potentially can be enrolled on those trials. And also because they want them in line so that if they don't respond to the next line of salvage chemotherapy, you're not then waiting a month or two before they can ultimately get treated. So we are actually probably seeing patients referred in earlier, and as a result, their disease is under slightly better control than if they're just found out that they were growing through second-line therapy and get in to see you a week or two later.
Sure.
Yeah.
And the impression seems to be that the better the patient coming in, the more likelihood you are to successfully get the patient through.
But in the real world, as you mentioned, right, that 50% of patients who are getting on commercial CAR T cells wouldn't have qualified for the clinical trials, right? Mm-hmm. So we're getting a really mixed bag of different things, which is interesting to watch. We'll see what will come out of it, if the high response rate will be maintained, or the safety will be maintained in these patients.
Yeah. I think that we'll hear this data soon, and I think that we're able to get these 50% of patients that weren't eligible for clinical trials safely through. It's not just about organ function and performance status, but it's also about histologies, right? These clinical trials enrolled patients with specific histologies, but people are referring people in now with transformed marginal zone lymphoma, with transformation from CLL, and these are these lymphomas don't seem to be exactly the same, and I think that may actually impair some of the real-world efficacy results that we see.
Now, one of the things I was really impressed by in your session was having a neurologist there. It's very unusual for us to have a non-oncologist come to the meeting, but I thought she actually contributed enormously to being there. And I was also struck by just incredibly how knowledgeable about not just the neurology of the CAR T cells, but the patients that she's treating that she was. It was actually very impressive. So it's nice to obviously be thinking about how you develop a really skilled multidisciplinary team to manage a patient through this toxicity.
Yeah, it's not different than for allotransplant. Yeah. It's just the specialists are slightly different.
Yeah, sure.
At the Farber, we also have a neurointensivist who's taken an interest in this, and he's going to publish his own publication about what he's seen in our patients. And then we also we've seen some cardiology toxicity related to the cytokine release. So we have a cardiologist who's been following these patients, and they're all very interested in doing their own research and trying to figure out preventative mechanisms or screening mechanisms to minimize toxicity. So it's like allotransplant, just a different group of specialists.
Well, it sounds like it's like allotransplant, but a little bit better, because what I picked up from that session was, although the neurotoxicity shouldn't be diminished, what everyone says was that we've got a better handle on how to do it, and it is self-limiting and is reversible.
Yeah, I think whenever you're trying to talk to someone about an allotransplant, it's the unknown risk that you don't know what to tell them. You don't know what to tell them that they're going to be like 6 months after the allotransplant, and we have a pretty good idea of, we're able to pretty confidently tell someone what they're going to be like 6 months after CAR T cells, and it's going to be very similar to before they got sick, as long as they respond well and have their disease improve.
So there we have it. So yet again, at the end of the second day, I think you can pick up from us the excitement we felt at the three, I think, pretty wonderful sessions that we had today. And that's iwNHL 2018 over, and we look forward to seeing you all next year in Cambridge, Massachusetts for iwNHL 2019.